Good morning, good afternoon, and good evening, everyone. And welcome to our webinar titled Old Dogs Benefit from New Tricks, 3D Analysis of Arterial Injury to Study Nrf2 Dependent Inhibition of Neointimal Hyperplasia. This is Liam Sanyo, and I'm very pleased to be your host for today's event. This program was developed through a collaboration between the American Physiological Society and Miltenyi Biotech in support of their shared mission to provide education and value to researchers everywhere. We're very fortunate to have Edward Moreira Bahnson here with us today, who is an assistant professor in the Department of Cell Biology and Physiology at the University of North Carolina at Chapel Hill. Today, Professor Bahnson will be presenting the development and validation of a methodological approach using light sheet fluorescence microscopy for the imaging of preclinical models of vascular injury with a focus on minimizing analysis bias while increasing precision and reproducibility. And with that, I'm very pleased to hand things over to Professor Edward Bahnson. Professor Bahnson, thanks so much for joining us today, and the floor is yours whenever you're ready.
Hello, I am Edward Moreira Bahnson, and I'd like to start by thanking the American Physiological Society and Miltenyi Biotech for organizing this webinar series. Today, I'm going to tell you how activation of Nrf2 inhibits neointimal hyperplasia and how we use 3D analysis instead of traditional 2D histology to study this phenomenon. Atherosclerosis is the leading cause of death and disability in the US. Moreover, it's actually a leading cause of death worldwide. This diagram shows to the left how a normal artery looks, and towards the right we show progression of atherosclerosis, which is a chronic inflammatory disease characterized by subintimal deposition of lipids. This atherosclerotic plaque can become eventually unstable, and if it ruptures and causes thrombosis, can lead to myocardial infarction and or stroke. So it's incredible that 1 every 3 deaths in the United States is directly attributed to cardiovascular disease. And that results in a staggering number to me, that is 1 death every 40 seconds from cardiovascular disease in this country. Moreover, because of an aging population, it's only expected an increased prevalence of both coronary and peripheral artery disease. That increase is observable already in that from 2000 to 2010, there's been a 28% increase in the total number of inpatient cardiovascular interventions. What are these cardiovascular interventions? Well, they are the treatment options that we have for severe atherosclerotic disease. They include, but are not limited to, angioplasty with or without stenting, endarterectomy, and surgical bypass grafting. All of these procedures aimed at reestablishing correct blood flow, and they're very good at it, at least in the short term. However, the long-term success of these procedures is hindered by the development of restenosis. Restenosis is the recurrent luminal narrowing, which is the product of the arterial injury response, not the underlying pathology that led that patient to get the revascularization. These recurrent luminal narrowing is driven by 2 processes: neointimal hyperplasia, which is the proliferation and migration of smooth muscle cells and adventitial fibroblasts into the intima, and vessel remodeling. Here we have 2 optical coherence tomography pictures showing human coronaries. To the left, we see a healthy coronary where you can appreciate a patent lumen and the 3 layers of the artery, adventitia media and intima. And to the right, we see in-stent restenosis 9 months after stent implantation. Here, these bright lines are the struts of the stent, and inside you can see this concentric hyperplastic lesion with a narrowed lumen. In the lab, we have different models to study this. My lab studies the rat carotid balloon injury model, which is what I refer to as an old dog because it's been around for a long time. This model involves isolating the left carotid of the rat. Here the heart is down and the head of the rat would be up. So we identify the common carotid artery, the internal carotid artery, external carotid artery, and superior thyroid artery. we suture the external and the superior thyroid and clamp the common carotid artery and the internal carotid artery for control. So at the bifurcation between the superior thyroid and external carotid artery, we perform an arteriotomy, and then we place and insert a balloon catheter and advance it to the common carotid. There we inflate it to 5 atmospheres, and we leave it in place for 5 minutes. Then we remove it and we ligate the external carotid at the site of the bifurcation with the internal. We remove the clamps to reestablish blood flow, and at this point we can apply a therapeutic in the lab, and all the data that I'm going to show you today involves application of a therapeutic peri-adventitially dissolved in a gel, pluronic gel, for local delivery of a therapeutic. Then we close the incision and we can study what happens 1 day, 3 days, 2 days, or a month after the injury. So this is how a normal rat carotid artery looks by histology. This is stained by H&E. You can see a patent lumen and the media, a single cell layer of the intima. And 2 weeks after balloon injury, this is how it looks by histology. You can see a thickened media and this hyperplastic lesion with a narrowed lumen. I told you in the title that in the lab we study this by 3D, and I'm going to go into detail for later in the presentation. But here I'm showing you that this is a longitudinal view of a common carotid, and here's a bifurcation of external and internal. And we can see the hyperplastic lesion here, and this is a cross-section where you see the same thing. There is abundant evidence in the literature suggesting that restenosis is driven by redox dysfunction, where vascular injury leads to activation of NADPH oxidase 1 or NOX1. This enzyme produces superoxide and later hydrogen peroxide, increasing these levels of these reactive oxygen species, which will signal smooth muscle cells to migrate and proliferate, resulting in neointimal hyperplasia. Because redox dysfunction drives neointimal hyperplasia, we chose a target that affects the redox status of cells. Which target? Nrf2. Nrf2 is a transcription factor that under basal conditions is bound in the cytosol to its inhibitor repressor KEAP1, which is a part of an E3 complex catalyzing the ubiquitination of Nrf2, targeting it for proteasomal degradation. In the presence of electrophiles, electrophilic stress, therapeutic electrophiles, or oxidative stress, KEAP1 has cysteine residues that sense these electrophiles or reactive oxygen species that induces a conformational change in KEAP1 that doesn't allow for ubiquitination of Nrf2, and therefore no degradation of Nrf2. Then the newly synthesized Nrf2 can accumulate in the cytosol, translocate to the nucleus where it binds the antioxidant response elements, driving the transcription of antioxidant enzymes and other cytoprotective genes. So our hypothesis is that an electrophile can be used to activate Nrf2, increase the cell's antioxidant enzymes, therefore decreasing the levels of reactive oxygen species, inhibiting the whole process that leads to neointimal hyperplasia. So the data that I'm going to show today, both for Nrf2 mediated inhibition of hyperplasia and the development of a 3D method to analyze this phenomena, is been part of my first graduate student's thesis. Almost Dr. Nick Buglak. He's writing his dissertation and hopes to defend next semester. So what electrophile we chose? First, we started working with cinnamic aldehyde, which is a natural product present in cinnamon. Cinnamic aldehyde is an alpha, beta, unsaturated carbonyl, which results in this beta carbon being electrophilic, therefore capable of reacting with KEAP1 and activating Nrf2. So the first piece of data that I'm going to show you are smooth muscle cells stained for immunofluorescence, stained for Nrf2 in green. You can see that under basal conditions, this green staining is diffuse in the cytosol. But when we treat with electrophiles, cinnamic aldehyde at 100 micromolar or a control sulforaphane that is also a known activator of Nrf2, we see translocation of that signal and accumulation in the nucleus. Another way to look at this is by a Western blot of subcellular fraction, where the nuclear fraction, the Nrf2 signal is increased upon treatment with sulforaphane or cinnamic aldehyde. So this tells us that Nrf2 actually goes to the nucleus, but does it activate the downstream pathway? First thing we looked at is a canonical transcriptional target of Nrf2, hemoxygenase 1 by Western blot. We see that treatment with cinnamic aldehyde induces and increases the level of this protein. After we knew that we were activating the pathway with CA, we chose to look at other relevant antioxidant enzymes. Superoxide dismutase 1, that detoxifies superoxide anion. GCLC involved in the synthetic pathway to glutathione, and peroxyredoxin 1, which detoxifies hydrogen peroxide. So we saw that treatment with CA induces an increase in all these three enzymes, SOD1, GCLC, and peroxyredoxin 1. But not only does it increase the levels of the proteins, does it actually affect their activity? First thing we looked is at SOD activity in smooth muscle cell extracts treated with cinnamic aldehyde, and we see that there's a significant increase in superoxide activity upon treatment with CA. When we looked at the levels of glutathiones that should be affected by an increase in GCLC, we also saw that cinnamic aldehyde induces a significant increase in glutathione levels, suggesting that with CA we're inducing the Nrf2 pathway, increasing the levels of these antioxidant proteins, and affecting the redox environment in the cell. This should lead to a change in the proliferation and migration of smooth muscle cells. The first thing that we looked at was an MTT to see that actually, in fact, cinnamic aldehyde was inhibiting cell proliferation. And MTT, we used it as a first surrogate to explore what concentrations we had to work with. And we found an EC50 a little above 100 micromolar. But we didn't know, of course, if this was inhibiting proliferation or was inducing cell death, because cinnamic aldehyde is a reactive aldehyde with the potential for toxicity. So we decided to assess apoptosis. And first, we see that cells stimulated with PDGF have the majority of cells are alive, and 100 micromolar of cinnamic aldehyde significantly reduces the number of live cells without an increase in death. Here, the white, gray, and red are early apoptosis, late apoptosis, and dead cells, and there is no difference, statistically significant difference in these dying or dead cells compared to control, but there is a significant reduction in viable cells. This suggests that cinnamic aldehyde reduces cell number without inducing death, therefore inhibiting smooth muscle cell proliferation. Moreover, when we actually treat with high concentrations of cinnamic aldehyde, increase in cell death, because at 1 millimolar, cinnamic aldehyde is being toxic towards these smooth muscle cells. The other thing that we looked at was a migration of smooth muscle cells. To do this, we used the scratch wound assay. Cells are plated, then a scratch wound is performed with a pipette tip, and when cells are not stimulated, they just don't move and don't migrate and don't proliferate. When we stimulate the cells with PDGF, cells migrate towards the wound. And treated with CA, they don't. This is quantified in this graph over here, where you see that cinnamic aldehyde statistically significant reduces the number of cells that migrate towards the scratch wound compared to PDGF alone. We wanted to see if this effects of activating Nrf2, affecting the redox status, and inhibiting cell proliferation also happened in vivo. The first thing we did was perform the carotid balloon injury, apply periappendicularly cinnamic aldehyde, and look by immunofluorescence if there was translocation of Nrf2 to the nucleus. After injury, when we treated with the vehicle alone, we see in red here staining for Nrf2, and you see a very diffuse and not localized red staining in the vessel wall. Here in green, we see the autofluorescence of the different lamella, and L is the lumen So this is the vessel of the rat carotid. When we treat with cinnamic aldehyde, 100 micromolar cinnamic aldehyde periadventitially, 3 days after balloon injury, we see this intense red fluorescence that overlaps with the blue of the nucleus, both in the adventitia and in the media, suggesting that there is activation of Nrf2 in vivo as well. So what if this is true, are we actually affecting the redox environment of the vascular wall after arterial injury? The first marker of a redox dysfunction or oxidative stress that we looked at is 3-nitrotyrosine, that could be seen as a fingerprint of peroxynitrite production. We can see first and very clearly that an uninjured vessel doesn't have a lot of staining, but after balloon injury, there's a significant increase in fluorescence for 3-nitrotyrosine that is significantly decreased in the presence of cinnamic aldehyde. This is quantified here and normalized to number of cells counting DAPI nuclei, and we see a significant decrease in 3-nitrotyrosine. The second thing that we looked at was levels of DHE fluorescence, which indicates levels of superoxide. So superoxide reacts with DHE, forming 2-hydroxyethidium, which is a fluorescent product that correlates with superoxide levels. Again, the first thing that we see is that injury, there's a significant increase in red fluorescence in the arterial wall compared to uninjured vessels. And this is expected because we know that vascular injury results in activation of NADPH oxidase 1, which produces superoxide. And when we treat the arteries with cinnamic aldehyde, we see a significant decrease in red fluorescence signal normalized by cells. Other aspects to study were the infiltration of inflammatory cells, because after injury we know that neointimal hyperplasia is driven not just by the redox dysfunction of smooth muscle cells, but pro-inflammatory cells will come and produce reactive species in the vascular wall as well. So first we looked at neutrophils, assessing myeloperoxidase positive cells. And again, we see that there's an increase in signal after injury that is significantly inhibited with CA. So there's less infiltration of neutrophils. Then we looked at macrophages, looking at CD68 positive cells. And again, 3 days after injury, we see a significant increase in CD68 positive signal in the vascular wall, and that is significantly inhibited by treatment with 100 micromolar of CA. We have an activation of the pathway in vivo, decrease in oxidative stress markers, and decrease in inflammation. Does this result in a decreased proliferation and hence decreased neointimal hyperplasia? To look at that, first we assessed the proliferative index of the vascular walls, treating the rats before sacrifice with a BRDU, and then counting BRDU positive cells over total number of cells to come up with this proliferative index. That index is increased, of course. It's close to 0 in quiescent uninjured vessels, but after 3 days after injury, we see an increase in proliferation, which is expected and well described in the literature. But treatment with CA decreases the level of the proliferative index, resulting in a decrease in neointimal hyperplasia 2 weeks after injury. We can see this by histology in these cross sections, where we see the neointimal hyperplasia that is inhibited in CA-treated animals. We can quantify that with these morphometric parameters: intima to media ratio, which is, as it is, the ratio in the area of the neointima and the media, or percent occlusion, which is the percent of the cross-section of the lumen that is occluded with neointimal hyperplasia. Both parameters are significantly inhibited by treatment of cinnamic aldehyde. So the working model that we have is that cinnamic aldehyde is both inhibiting neointimal hyperplasia and activating Nrf2, which should inhibit reactive oxygen species and inhibit neointimal hyperplasia. However, we don't know if there are Nrf2 independent mechanisms at play. To summarize, CA treatment leads to nuclear localization of Nrf2 and activation of the pathway, both in vitro and in vivo. The cinnamic aldehyde treatment inhibits smooth muscle cell proliferation and migration in vitro, and decreases levels of redox markers both in vitro and in vivo. Most importantly, cinnamic aldehyde treatment prevents neointimal hyperplasia in vivo. So the hypothesis is that this inhibition of neointimal hyperplasia is Nrf2 dependent. So to assess if the effect of cinnamic aldehyde at inhibiting neointimal hyperplasia is Nrf2 dependent, we turned to a Nrf2 knockout rat colony that we have in the lab, and we will compare the effect of cinnamic aldehyde in wild-type and Nrf2 knockout animals. We isolate smooth muscle cells from the aorta of these animals, and we're going to study the effect on proliferation and migration of these different genotypes. So the first thing, we repeated the apoptosis assay in viable cells, but comparing what happens in wild-type versus knockout animals. And as expected, in wild-type animals, I had shown you before, cinnamic aldehyde reduces the number of viable cells, and that reduction does not happen in knockout cells, suggesting that the inhibition of cell proliferation is Nrf2 dependent. We again looked at cell death to make sure that there was no death induction at 100 micromolar, and again, there is no increase in cell death at 100 micromolar in either of these genotypes, suggesting again then that CA reduces smooth muscle cell proliferation in a Nrf2 dependent manner. The other component is migration. So we performed the scratch wound assay with smooth muscle cells of these two genotypes, and as we had seen before with a different strain of rat, cinnamic aldehyde inhibits migratory smooth muscle cells in Nrf2 wild-type rats. However, that inhibition disappears when we are looking at knockout rats, suggesting that this inhibition in migration is Nrf2 dependent. We wanted to dig a little bit further into this effect, and to do that, we wanted to see polarization of the cells towards the movement, in directional movement. To do that, we quantified the position of the centrosome in the cell. We stained the cells with tub GCP2 to find the centrosome, which are these little dots here. And we quantified if the centrosome was forward-facing towards the scratch wound, middle or rearward-facing. Here we have in yellow the cells with forward-facing centrosomes, and we see that cinnamic aldehyde reduces the number of cells forward with the centrosome that's forward-facing in the wild type and not the knockout. But we are going to quantify this to show you this better. So here we see how the number of cells with centrosomes towards the scratch wound is significantly reduced in wild-type cells, but not in knockout cells. In wild-type cells, actually, it looks like unstimulated cells that do not migrate. Another way to look at this is as the distribution of frontward-facing, middle, or rearward-facing centrosomes. In the wild-type animals, we see that when we stimulate with PDGF, the majority of cells have scratch or forward-facing centrosome. And the unstimulated cells actually have a pretty even one-third, one-third, one-third of cells, which indicates random distribution of the centrosome. The cinnamic aldehyde-treated cells also resemble these unstimulated cells with random distribution, a third forward, a third in the middle, and a third rearward facing. However, in the knockout animals, the distribution of centrosomes in the cinnamic aldehyde-treated animals resemble the stimulated ones, with the majority of the cells having a centrosome forward-facing or towards the scratch one, suggesting that this polarization is induced by CA is the inhibition of the polarization induced by CA is Nrf2 dependent. We went back to assess levels of superoxide by looking at the fluorescence of DHE that we had seen before was decreased by cinnamic aldehyde. And we did this assay in vitro with wild-type and knockout cells. We see that treatment with PDGF increases fluorescence per cell, and treatment of cinnamic aldehyde actually decreases it in both wild-type and knockout. However, the decrease in DHE fluorescence by cinnamic aldehyde in wild-type animals is more pronounced than what happens in knockout cells. So it inhibits this DHE signal to a greater extent in wild-type animals. So this suggests that there could be a non-Nrf2 dependent mechanism by which cinnamic aldehyde is resulting in less superoxide levels as well in knockout animals, but not enough to see an effect in proliferation or migration. So what happens in vivo, right? This is all a comparison of wild-type and knockout animals in vitro, and I have shown you that by traditional histology, cinnamic aldehyde inhibited neointimal hyperplasia in Nrf2 wild-type animals. So before I tell you what happens now, I'm going to take a little make a little parenthesis and tell you about this new methodology that we developed to study neointimal hyperplasia by 3D. Why did we do this? Well, first, 2D datasets have intrinsically less data than 3D datasets, so we are missing potentially data that could be useful. No matter how many methods you have in place to minimize user bias, in 2D you have to select the slides that are going to be quantified or included in the quantitative analysis, and that is impossible to completely avoid user bias. It relies on physical sectioning. If you've done this yourself, you know how time-consuming and mind-numbing it is. Actually, it's completely intellectually not engaging. And I believe that it's very difficult to assess remodeling in 2D. Why? Because there's this bias introduced by how you cut. If you have an artery and the blades have to be perfectly perpendicular to be able to assess perimeter and radius and cross-sectional area. And if you deviate from that angle and you cut at an angle compared to the plane of the artery, then the perimeter and the radii are going to be artifactually modified without really having necessarily be a change in vascular remodeling. And finally, I think that we can analyze more parameters in 3D that might lead to scientific discovery potentially. And a problem that I think is huge in the field is the lack of standardization. The field recognizes there is a lack of rigor and reproducibility in studies of restenosis in these animal models of arterial injury. So when you go to the literature and you look at how people analyze that, first, the number of sections used for analysis vary significantly from 3 to 10. And secondly, when you go to look for the description of how these 3 to 10 cross-sections are selected to be analyzed, you find some papers that actually don't state anything, some papers that do state, but to a different degree of detail in the description. And I think that is a major contributor to this lack of reproducibility. So what was our answer to this issue? So we decided to use a 3D imaging methodology, light sheet fluorescent microscopy, to analyze arterial injury. This involves clearing the tissue that we're going to analyze. We embed it in agarose. You place that in a sample holder, and it goes to the microscope, where a light of sheet is shown to clear tissue sample in a 90-degree angle with the plane of detection. This is a second project of my graduate student Nick Bublek. Here I'm showing you how a rat carotid artery looks before clearing. We embed this in agarose just because it's easier for us to handle, especially after clearing this small artery. It's very difficult to see and we can lose it. So in the agarose it's easier to manipulate. And you can see that after clearing, or better said, you cannot see how this whole block becomes clear. At a higher magnification here, you can see that the artery is still there. We use iDisco Plus as the method for clearing, as I mentioned before. It involves a series of dehydration steps using methanol, and then clearing with organic solvents, and it ends in DBE as the final solvent where it's going to be imaged. The first thing we did is let's try and see if this works. We took a healthy carotid artery and ex vivo we incubated with an antibody anti-CD31 to label the endothelial cells and mark the intima of the vessel. and we imaged to see if the resolution was enough to see what we wanted to see. And voila, I'm showing you here this beautiful image of a rat carotid where you can see a single cell layer of CD31 positive cells sitting on top of the media that is visible due to its autofluorescence, and you can even appreciate the individual lamella of the media. This Z-stack of these images can be used to render it in 3D, where you can see this beautiful lining of the inner space of the vessel with CD31 positive cells, and here the media with its autofluorescence. We got really excited that we could see this, so immediately I told Nick, well, we have to see if we can see an injured artery and see a stenosis and neointimal hyperplasia. I'm going to show you a video here. Take a look at this space here where you'll see neointimal hyperplasia. There it is, and a narrowed lumen is going to come back so you can see again. And then it's going to highlight where the site of stenosis is and the neointimal hyperplasia. This was so beautiful that immediately we decided to actually come up with a method to quantify stenosis using LSFM. To do that, we came up with this workflow using the software Imaris. We import the Z-stack into Imaris, and using a tool called Volumes, we encapsulate these, the tool is called Surfaces. We, using the CD31 fluorescence and the fluorescence of the internal and external elastic lamina, we can use those surfaces to encapsulate different volumes: the volume of the media, the volume of hyperplasia, and the volume of the lumen. Doing that, we can see the entire vessel and identify where the site of stenosis is. So here the green is the internal elastic lamina enclosed volume, and the white is the CD31 enclosed volume. So you see a pinching in the lumen there, which is the site of stenosis. Where the artery has not been injured, like here, you see that the white, the CD31 positive, actually overlaps with the green, the internal elastic lamina, because a healthy artery, the endothelial cells actually sit on top of the internal elastic lamina, and the noise of the method actually sees that sometimes one looks a little bit above the other, but it's basically they're sitting on top of the other. So as you can see here, you don't have to select this cross-section or this cross-section to show your data. You can show the whole artery and showing the total site of stenosis. However, still, selection of the region of interest where injury occurs, if you were to select that yourself by looking, it would be subject to some user bias. So we decided to come up with a method to minimize that and automate this process of selecting where does injury start, where does injury end. To do that, we use the filament tool in Imaris to find the center of the artery and this distance render, where we create this heat map where each voxel is actually a distance in micrometer. With this, we can actually record all the radii along the entire length of the artery from that filament toward the intima, the internal elastic lamina, and the external elastic lamina. With all those radii, we can trace these plots, where 0, 0, the origin here is the bifurcation between the internal and external carotid, and the distance here is into the common carotid. And we can see that the difference between the intima and the internal elastic lamina would be the average thickness of neointima at that particular distance. So calculating this delta radius, we can create this other plot where you can see actually the topography of neointimal hyperplasia and see where there is lesion. But how big that delta radius need to be for it to be considered neointimal hyperplasia? To minimize user bias in that decision, we do the same delta radius in a healthy contralateral artery from the same rat. The right artery wasn't injured, and we also calculate these radii, and we trace the plot. And as expected, the radii for the healthy artery is around 0 because, as I mentioned before, the intima sits on top of the internal elastic lamina. With all that data, we create a 99% confidence interval, and we define that as the threshold above which we determine that's hyperplasia, and below that it's a healthy artery. So we know exactly the length of injury, and that is the length of the artery that we're going to use to calculate all the morphometric parameters, like getting this media volume, getting the intima volume, and be able to actually calculate an intima to media ratio, as I have mentioned before. So we needed to compare that with traditional I to M, and so we took a separate cohort of rats and we calculated the intima to media ratio by histology staining with H&E, and we compare that with the intima to media ratio calculated by optical volume in arteries analyzed exclusively by LSFM. And of course we see here that there is no difference, indicating to us that the parameter that we can calculate by LSFM is actually the same parameter that has been used and validated in the literature for decades. However, if you take a closer look and we decide to measure the precision of the method by calculating the coefficient of variation percent, we see that the optical volume has a smaller CV percent compared to the H&E, suggesting that this measurement is actually more precise. As a side note, I know that that's rat, but a lot of you might work with mice, so I wanted just to show you that we can use LSFM in mice models, both of restenosis and atherosclerosis. Here I'm showing you the aortic arch of an ApoE knockout mouse that has been fed a high-fat diet for 14 weeks, and you can see in the arch in red the atherosclerotic plaque. And here you see results from the carotid ligation, the mouse carotid ligation, and the comparison between traditional histology and LSFM to assess neointimal development and I to M ratio. Again, like in the rat, you can see this lumen that is lined with CD31 positive cells, and you can see the longitudinal and cross sections where you can appreciate the development of neointimal hyperplasia. When we quantify and compare optical volume and traditional histology, again, we find that the I to M parameter that's being validated in the literature is no different by optical volume. And again, the precision is improved, even though this model is way less precise than the rat model. So going back then to our question, we went to the in vivo model using the Nrf2 wild type and knockout rat. We performed the balloon injury, and we apply either cinnamic aldehyde dissolved in chloronic or vehicle alone, and then at 2 weeks we obtain the arteries, we clear it and image it by LSFM. So the first thing that I'm showing you is the comparison of this wild type and knockout rats at baseline without any treatment of cinnamic aldehyde. We needed to know that the knockout rat, which has not been used for this model before, to our knowledge, that it actually behaves the same at baseline. And as you can see here, the artery occlusion that we induce and the I to M ratio that we induce with balloon injury is the same in the wild type and the knockout rats at baseline. Knowing that without treatment, these rats behave the same in these morphometric parameters, we looked at what happened when we treated with cinnamic aldehyde. Here I'm showing you representative images of total neointima volume. As the previous slide suggested, when there's vehicle alone, wild type and knockout have basically the same neointimal hyperplasia development. And when we treat with cinnamic aldehyde, as expected, the wild type shows a significant decrease in the volume, but the knockout rat does not appear, cinnamic aldehyde does not appear to have a significant effect. A different way to look at this is these radii plots. When we plot the delta radius for wild type and Nrf2 knockout rats in the vehicle treated animals, we see that both have this injury. The area under the curve is proportional to the amount of neointimal hyperplasia, and we see that when we treat with cinnamic aldehyde, that area under the curve is significantly reduced in the wild type rat, but not so much in the Nrf2 knockout. Of course, we need to quantify this and do some stats to know that this is true, and this is what we did. Different parameters, arterial occlusion percent and hyperplasia volume, both parameters are significantly reduced in the wild type when we treat with cinnamic aldehyde, but not reduced in the knockout animal when we treat with cinnamic aldehyde. So one thing to consider here is that hyperplasia volume here is all the length that has been injured. One way to normalize that is to divide that volume by the extent of injury, and that is the average area of cross-section of neointimal hyperplasia. And when we normalize for that, this difference is maintained with a decrease in hyperplasia only in the Cinnamic aldehyde-treated wild-type animals, but not knockout animals. So this suggests that the development of neointimal hyperplasia is inhibited by CA in a Nrf2 dependent manner. All these are volumetric analysis, but we have all this data with the radius. So as I said, we can analyze more parameters. The average thickness of hyperplasia is also inhibited by CA only in the wild type and not in the knockout. But importantly, the peak injury, which is the site of more stenosis along the artery, is of significance clinically, because even if you have the entire artery injured but with very little hyperplasia, that is not as serious as if you have a site where it's really occluded. So we looked at peak occlusion with the delta radius, and that peak occlusion is also inhibited only in wild-type animals treated with CA. Moreover, another very important parameter is the radius of the lumen, that if your treatment of interest works, should keep a bigger and more patent lumen. So here we are looking at an increase in lumen radius, and that only is observed in wild-type animals treated with Cinnamic aldehyde. So as I mentioned before, neointimal hyperplasia is the main driver of restenosis, but not the only driver of restenosis, even though there's some debate in the literature about the contribution of remodeling. But remodeling happens after arterial injury, and we have two types of remodeling happening: compensatory or positive remodeling, and a constrictive remodeling. So in terms of compensatory remodeling, once the lumen, the cross-section of the lumen starts to decrease due to formation of neointimal hyperplasia, the vessel reacts by expanding outwards to compensate and try to maintain that lumen cross-section. So what we saw when we looked at that is that Cinnamic aldehyde induces positive remodeling. That is a delta of the radius to the external elastic lamina, comparing the injured artery with its contralateral artery that has not been remodeled for sure. So an increase in that radius is indicative of compensatory remodeling. And there is a significant increase in CA-treated animals. In the wild-type animals, there's this increase that is not observed in the knockouts. Moreover, the knockouts seem to actually, at baseline, do a little bit of better at remodeling than even without any treatment. But if you compare the wild-type animals without treatment and with treatment, it seems like Cinnamic aldehyde promotes compensatory remodeling, where in the knockout animals, maybe because they already remodel even without Cinnamic aldehyde, there is not a significant effect. The other side of the remodeling coin, as I mentioned before, is the constrictive remodeling. It's characterized by loss of elasticity of the vessel, fibrosis, and deposition of collagen. A way to assess this by traditional histology is to look at the amount of collagen. But since we have all this radii, actually we can see the thickness of the vessel and see if it actually gets thicker. And so two ways to analyze this was the constrictive remodeling as being the change in thickness, and we see that there is no effect apparently in compared to pluronic alone and Cinnamic aldehyde. This there's a statistical significance there, but there's no significance between wild type treated with CA and not treated with CA, or knockout treated or not treated with CA. So even though there is this difference there, there is no evidence suggesting that CA actually influences negative remodeling in a Nrf2 dependent or independent manner. Another way to look at that it may be more clear, is the normalized media area, so the average area of the media that it's supposed to thicken with constrictive remodeling. and there's no difference in any of the conditions, suggesting that neither CA in either genotype is affecting the constrictive remodeling, that it exists. And we can see that it exists because it's this delta medial thickness compared to the contralateral artery is greater than 0, meaning that the media in the injured artery is thicker than in the contralateral in all points. I hope that I've shown you that CA inhibits smooth muscle cell polarization and migration in a Nrf2 dependent manner, that it inhibits neointimal hyperplasia and promotes compensatory remodeling in a Nrf2 dependent manner. But importantly, that LSFM allows for these 3D analysis of arterial injury models, not just the rat, but also mouse. And with this new tool, the morphological analysis is more precise, and the estimates are actually comparable to 2D. So the I2M does not really change. However, we reduced user bias with 3D analysis. It allows for both volumetric and aerial analysis. It allows you for accurate remodeling quantification. And I think that it can drive discovery like this new data that CA appears to not influence negative remodeling, but appears to influence compensatory remodeling in a Nrf2 dependent manner. With that, I'd like to thank the lab. These are all the people that actually do a lot of the work. And our collaborators, Dr. Pablo Ariel, who's an expert in light sheet fluorescent microscopy, Dr. Francis Miller, Dr. Jennifer Lucidi, and of course the sources of funding. I am funded by a K award from the NHLBI, and my student Nick is funded by the American Heart Association predoctoral fellowship. With that, I'd be happy to answer any questions.
All right, fantastic. Thanks so much, Dr. Bahnson, for the really fantastic presentation. All right, so just jumping in here. First question, have you examined animals with smooth muscle-specific Nrf2 knockout? It looks like the drug treatment and the knockout rat was a global knockout.
Yeah, that is a great question, and unfortunately, we do not have access. There's no rat available that is smooth muscle-specific knockout, so we only have access to a global knockout. But, you know, that is a question that we do have in mind, but we cannot answer right now, unfortunately.
Fantastic. All right, next question here. This one was about flow and resistance for the compensatory. Here, we'll go back to this slide here. That should appear. Yeah, with the compensatory remodeling, did you look at flow and resistance? Was there any change in flow and resistance?
That's a great question as well. We did not look at parameters in vivo, so we do not know that. But it's, of course, changes in flow is going to drive remodeling for sure.
Fantastic. All right, next question here. Do you foresee cinnamic aldehyde being applied to atherosclerotic vessels in future applications to reduce reactive oxygen species and growth of the atherosclerotic plaques?
So that's also very interesting. So cinnamic aldehyde for us was a modeled molecule that we do not foresee has a very a bright future in translation, because as somebody else pointed out in the Q&A panel, we have to use 100 micromolar to induce Nrf2. In the lab now, we are exploring for atherosclerosis treatment a nanomolar activator of Nrf2, CDDO methyl or bardoxolone methyl, which is a nanomolar activator, and also it's actually undergoing clinical trials for different applications. So there is a future for human use of this drug, and we are actually encapsulating this drug in nanoparticles that we target to atherosclerotic plaque. We have evidence that with this treatment with CDDO-methyl, we can activate Nrf2 in atherosclerotic arch, in the arch of atherosclerotic animals. And we are at the moment performing in the middle of a study to see if there is inhibition of plaque progression or not.
Fantastic,
I have
great
to
answer.
point out something else regarding this question. The literature is a little bit controversial about the role of Nrf2 in atherosclerosis. There's some evidence of pro-atherogenic role of Nrf2 activation in late stage atherosclerosis, but that is with global knockouts. With every study that has used an activator that actually pushes the Nrf2 activation that's already there to more, all those studies have shown anti-atherosclerotic effects of Nrf2 activators.
Great answer. All right, so yeah, going back to that question that you mentioned before, the question was, so you use 100 micromolar, which is rather high. Why do you use so much? Does this cause issues?
Well, so we use that much because we tested what concentration was actually capable of inhibiting vascular smooth muscle cell migration and proliferation in vitro. And, we did an MTT first and then scratch wound assays, and we needed 100 micromolar to activate Nrf2 and have a biological effect. That is why we use that much. That being said, we applied 100 microliters of 100 micromolar CA periadventitially. There's no systemic side effect because we do localized application. We're not injecting a solution into the animal. We apply this in a gel that acts as a depot for the drug on top of the artery that's been injured. So, but yes, as I mentioned before, we started this study to actually prove that Nrf2 activation could be a therapeutic target. Now we are moving on to better activators that target this pathway better with a more promising translational future.
Great answer. All right, next question here about the technology. How small of a vessel were you able to image in 3D? Did you try any peripheral vessels?
So that's a great question too. You see my mouse hovering. So these are
I do, yeah.
capillaries in a neovascularization, and this is angiogenesis in a mouse. So you see these capillaries are tiny. We are hoping to image a mouse leg next month in August to see actual collateral circulation in mouse. But I hope that this image here is very clear that we can see capillaries clearly stained with CD31, no problem. I'm going to stop share. So I am going to answer this NOX1 question. So Jacob says that I mentioned NOX1. On the other hand, NOX4 has been shown to be necessary for exercise-mediated improvements in obesity-induced vascular dysfunction. Would you expect indiscriminate quenching of ROS through Nrf2 activation in a therapeutic context, possibly leading to unintended vascular complications? So this is a very, very important question. The lab thinks that indiscriminate use of antioxidants, my philosophy to addressing antioxidant therapy, precisely thinks about these issues. We don't believe that global activation of or global scavenging of reactive oxygen species is the way to go. In order to do that, there's three things that we are doing to address that. These studies that I showed you that are proof of concept are with local application to not have global activation of Nrf2. So this is, I am not affecting the vasculature in the legs. I'm not affecting the vasculature that has not been injured because I am putting the activator at the site of angioplasty. However, that is not really a translational approach because most of these procedures in the clinic are percutaneous. That is, you get the catheter, for instance, inserted through the leg, and then you do an angioplasty in the coronary. So you don't have the artery exposed in open surgery to apply a medication, a depot, a gel locally. So what we do in the lab is targeted delivery of activators of Nrf2 using nanocarriers. And we develop these particles called arapas. There's one of the papers that describe our first iteration of arapas. We're working on a second and third generation now. ARAPA stands for antioxidant response activating nanoparticles. So we are aiming to preferentially accumulate the Nrf2 activator at the site of arterial inflammation, arterial injury, to not globally influence the redox status of the whole vascular system. And the third thing is the role of Nrf2 itself. This is not going to be a global scavenger that distributes in the cell. Different cells will activate the antioxidant response differently, and different enzymes are going to be targeted to different compartments. So we have to, of course, assess the potential problem with changing and modifying the physiological role of redox signaling. But for instance, in our models, catalase was not influenced, so hydrogen peroxide, that would be the main reactive species produced by NOx4, could potentially not be affected, even though we did have a change in peroxyredoxin. So I guess that the bottom line is the effect on NOx4 mediated signaling remains to be studied. However, the fact that we are activating Nrf2 locally to the site of interest puts my mind at ease that it's not going to be a global effect on signaling where it's needed and physiological. I hope that, Jacob, that answers your question more or less.
Fantastic. And I think in the interest of time, maybe we'll just have one last question here. Can this method be used for small vessels, for example, in the brain?
That has a very quick answer, yes. So we we can address the next one. There's vasculature in the brain with the LSFM. There's a lot of literature out there. So did you try to conduct an in vivo subchronic treatment versus post-injury treatment? So I think this is a very important question when we think of translation. Where and when is the most important time point to decide to treat? All the evidence that I've collected, starting as a postdoc and then in my own lab, tells me that the best time of intervention is the hyperacute phase after injury. And we actually right now, crossing fingers, we submitted an R01 proposal to study exactly that, if actually subchronic pre-injury will work anyway, and up to how many days post-injury we will see an effect. So a detailed timeline of when treatment would be most effective, I think, is paramount for a feasible translation to the clinic, because we need to know when patients are going to be prone to respond to treatment better. So thank you very much for that question, Frederick.
Yeah, fantastic. Well, thank you, Professor Bahnson, for all the really fantastic insights today. It's been a real pleasure having you with us.
Thank you very much. I enjoyed it, and I hope that our audience enjoyed it too.
Yeah, definitely. A big thanks, everyone, for joining us today for the webinar. And of course, we'd also like to give a big thank you to Miltenyi Biotech for sponsoring this event.
And one more thing, if you want to email me with a specific comment or question and want to go deeper into anything, my email is accessible online. Feel free to write to me, and I'd be happy to chat with you.
Fantastic. So in closing, thanks again for taking part in this Inside Scientific and American Physiological Society webinar, and we look forward to having you with us next time. Have a great day, everyone.